Hello everyone, welcome to Clarity for Sure. Today in this video we are going to discuss one of the important topics from your current affairs which you have been seeing in the news recently, that is the genome edited rice varieties that has been developed in India. We will be discussing about the basics of this genome editing and the technology that has been used in this particular genome edited rice, that is the CRISPR-Cas9 gene editing system, and we will also see some of the basic differences of that CRISPR-Cas9 system in itself. Okay, so without any further delay, let's get started. Now we will just quickly brush up the basics. We all know that in the last week India has released its first genome edited rice varieties known as Kamla, that is DRR Dhan 100 Kamla, which has been developed by the Indian Institute of Rice Research Institutes, Rice Research, which is located in Hyderabad. Next is your Pusa DST rice 1 variety that has been developed by the Indian Agricultural Research Institute of New Delhi. Okay, so what is the technology that has been used in this particular genome editing? The technology is known as CRISPR-Cas9 genome editing. Now coming to the specifics of the genome editing that has been done in this particular thing, we will just have a quick quick lookout of this particular genome editing that has been done in this particular rice, and in the future slides, in the further slides, we will be discussing about the exact process of CRISPR-Cas9 gene editing technology. Okay, what was the base variety from which this DRR Dhan 100, that is the Kamala, has been derived? It is the Samba Mashuri variety. The variety name is very important, which becomes important from your exam point of view only. And what is the method that has been used? The method that has been used is site directed nuclease 1, that is SD1. See, there are three genome editing methods: SDN1, SDN2 and SDN3. Do not worry, we will be discussing all these types at the end of this video after you gain a clarity on what a CRISPR-Cas9 technology is actually. Okay, the gene that has been targeted in this particular DRR Dhan 100 is cytokinin oxidase, also known as GN1A. This is very important. The target gene is cytokinin oxidase, which is also known as GN1A. Okay, and what is the purpose of this particular genome editing? This particular genome editing has been focused to enhance the grain number. That means to enhance the grain number, the paddy output. Okay, this is related to this DRR Dhan 100, that is your Kamala. Next, your Pusa DST rice 1. Here the base variety for this particular gene edited rice is Maruteru 1010. This is very important, and the method that has been applied here, the genome editing method that has been applied here, is also SDN1. Okay, that is site directed nuclease 1. Okay, here what was the targeted gene? The targeted gene here was the drought and salt tolerance gene. Okay, the purpose, the target gene itself tells us the purpose, that is to increase tolerance to drought and salinity. Okay, so these are all the important points which you have to note down regarding the base variety from which these two gene edited rice have been developed, and what method has been applied to this particular thing, and what was the targeted gene in these particular base varieties. So to to come up with the gene edited rice varieties. Now we have been talking about the gene editing methods, CRISPR-Cas9, everything. Now we will try to understand what this CRISPR-Cas9 actually is. Okay, this CRISPR-Cas9 is the abbreviation for clustered regularly interspaced short palindromic repeats. And this Cas9 is the protein associated with that technique, where this Cas9 protein is a powerful and a precise genome editing tool which actually allows the scientist to make the changes in the target DNA of a living organisms, including plants also. Okay, CRISPR-Cas9 is nothing but a genome editing tool where this Cas9 protein actually, that is the genome editing tool which acts like a scissor, and this Cas9 is actually guided by a guide RNA which guides that Cas9 to a specific DNA site where we have to make the changes to that particular gene so that the derived output which we are expecting can be achieved. Okay, this is about the basics of this CRISPR-Cas9. I hope the basics of CRISPR-Cas9 is clear to you. Now we will see how this actually works. Okay, now think this CRISPR-Cas9, this Cas9 protein as a pair of molecular scissors that is being guided by a GPS system. Here this GPS system is nothing but your guide RNA. What does this guide RNA mean? See, this guide RNA is nothing but a synthetic RNA. That it means it is a man-made RNA which actually guides this Cas9 units or Cas9 protein into a specific DNA site where the changes has to be made. Okay, from where this CRISPR-Cas9 technology has been taken out. See, in bacteria this CRISPR-Cas9 technology is inherently present to defend themselves against the viruses. Okay, this mechanism has been taken out from this bacteria and this has been applied to the genome editing tool. Okay, so this is the basics of this CRISPR-Cas9. So as I told you already, the scientists will use this guide RNA, that is known as gRNA, based on this CRISPR to find the specific DNA sequence in the genome. Okay, this Cas9, which I already told you, will act as a scissor-like enzyme that actually cuts the DNA at the target location identified by the guide RNA. Okay, once this guide RNA takes that CRISPR Cas9 protein to the specific site, the Cas9 protein will cut down the DNA sequence. The two strand will be cut off. Okay, once this DNA is cut, okay, the cells naturally try to repair it. Okay, and here where those three SDN1, SDN2 and SDN3 come into play. Okay, how? See, once this RNA is cut, the cell will naturally try to repair it. During this repair process, meanwhile in the process of repairing itself, scientists have variable options to modify that gene. Either they can disable the gene, uh, they can stop them from functioning, or they can edit the gene. In the sense they can enhance or modify its functions, be you know without applying any template. So now the question arises, what a template is. See, template is nothing but a customized design of DNA that can be inserted into this cut DNA so that a specific or a desired output can be achieved. This template will not be inserted in SDN model. Why? Because in SDN model the target specific location is cut off, so that gene has been disabled. So a desired trait has been achieved from that particular gene without placing any template. That is a modified gene. Okay, editing the gene, it means, what does editing mean? See, for example, in the DNA we know there are 4 molecules. So instead of A, the scientists can replace into G. Instead of T, the scientists can replace into T without inserting any new other foreign organism into that particular gene. the scientists will try to, insert the uh, actual DNA molecule that is being present in that particular organism. So that is your SDN2. What is SDN3 means? SDN3 is where the actual replacement of gene takes place, which is known as the genetically modified organisms, because we are going to introduce a foreign DNA into that particular DNA sequence which has been cut using this CRISPR-Cas9. So, to be very precise, SDN1 and SDN2 does not account to genetically modified organism, because there is no introduction of any foreign gene. Unlike SDN3, where a foreign gene is inserted or replaced, that gene is known as a template. Okay, a template has been inserted from a foreign organism. It is known as the genetically modified organism. Where the first two are not genetically modified, they are just edited. Their genes are just edited. I hope the difference between all the SDN1, SDN2 and SDN3 are clear, and now the CRISPR-Cas9 technology is also clear to you. Okay. Now showing a pictorial representation to you, as I was talking about the CRISPR-Cas9. Consider this as a DNA molecule, and this is a Cas9 protein, and a guided RNA. This yellow color is the guided RNA that actually takes this Cas9 protein to the specific site of DNA. This Cas9 protein then cuts this DNA complex which is double stranded. After this cutting is happened, the DNA tries to repair itself, and we have seen in the meantime of recovery what options the scientists are having. I hope this CRISPR-Cas9 technology is clear to you now. And this is another a pictorial representation where we were talking about the Cas9 protein and the guide RNA, and here is the custom design sequence. Okay, this sequence will be attached to this this target DNA. This guided sequence, which is the guide RNA, which is the custom design sequence, will take this Cas9 protein to this particular place. Okay, after this after this guide RNA guides this Cas9 protein into this particular site, the Cas9 protein will cut the target DNA. So the DNA is cut. The two-strand DNA is cut. Now in the editing, this is the place where the editing is done, either either through the genome, either through any of the three editing which we will be talking, or the DNA repairs itself in this particular section. I hope the working of CRISPR-Cas9 is clear to you, and how the scientist, after cutting the DNA, will be modifying or working in that particular specific site to achieve the desirable outcomes. Okay, I hope the basics of CRISPR-Cas9 and how does it work and the types of gene editing is clear to you now. Now coming to the exact gene editing methods that has been used by this used in this Samba Masuri rice and the other variety, Pusa variety. See here in this Samba Masuri, that is the Kamla variety, the CRISPR-Cas12 protein has been used. See, there are many proteins: CRISPR-Cas9, Cas10, Cas12, Cas12a, Cas13. There are variety of protein that can be used for this genome editing. But this CRISPR-Cas9 and CRISPR-Cas12 are the mostly or the widely used genome editing tools, which is more which is more powerful. And comparing this CRISPR-Cas9 and CRISPR-Cas12, this CRISPR-Cas12 protein is a little precise genome editing tool, which can be used as a molecular scissors to alter the gene function without adding any foreign DNA. Here, as I told you in the first slide, the target gene is cytokinin oxidase, that is GN1A. This cytokinin oxidase is one thing that actually controls the number of grains per panicle, and this cytokinin oxidase was one important was one important enzyme in that particular DNA, which actually played a negative role. That means it reduced the number of grain number per panicle. So what did scientists do? They used this CRISPR-Cas12 protein with a guide RNA, and this guide RNA took this particular CRISPR-Cas12 protein to that specific site of DNA where the cytokinin oxidase is present, and that enzyme has been cut off. So that enzyme has been cut off, and the DNA has been allowed to repair naturally without any intervention. So if there is no intervention, then it is known as the SDN1. Okay. Here, by targeting this particular gene, the scientists aim to increase the number of grains per panicle, thereby boosting the overall yield. Here I have already told you that the Cas9 enzyme was chosen for its ability to create a staggered cuts in the DNA, and this Cas9 protein, the enzyme has one important significance, where it leaves a sticky ends in the compared to Cas9. This Cas12 will leave a sticky ends on both the DNA sites, so that they can repair themselves naturally in a very less time compared to CRISPR-Cas9 technology. Okay, facilitating for more precise edits compared to the other CRISPR systems. Okay, so the main aim was to increase the grains per panicle. So the targeted gene has been cut off by the CRISPR-Cas12 editing in this particular Shamba Masuri rice. Okay, I hope this Kamla variety and the gene editing method that has been used in this Kamla variety is clear to you now. Coming to the next, that is your before going to the next, we will see the outcome. It has got high yielding output, 5.37 tonnes per hectare. I mean this Kamla variety, and potential it can go up to 9 tons per hectare, because the main idea of this gene editing is to what? Increase the number of grains per panicle, and it also showed the early maturity. Earlier it was 130 days to harvest. Now it is 15-20 days earlier than the original harvest. And what about the grain quality? The grain quality is same texture, same taste, and cooking quality of same Samba Masuri rice. I hope this CRISPR-Cas12 technology that has been administered to this Shamba Masuri rice and the outcome was Kamala variety is clear to you now. Now coming to the CRISPR-Cas9 editing that has been used in the Pusa DST rice one, we know the base variety was MTU 1010, and it is widely grown in 4 hectares in the southern, central and eastern India, and it is known for its high yield, early maturity, and it has a long slender grains. And genome editing here used was CRISPR-Cas9 and via SDN method only. The target gene was DST. We know that drought and salt tolerant. This editing enhanced the resistance to drought and salinity stress. Okay, and what was the outcome? 30.4% high yield in coastal saline conditions, 14.66% higher yield in alkaline conditions, and 9.67 higher yield in inland salinity. Okay. So this was the outcome of this particular CRISPR-Cas9 technology that has been taken place in the Pusa DST rice one. Okay. I hope the basics and the process in which CRISPR-Cas9 and CRISPR-Cas12 technology has been applied to this particular grains are clear to you now. So, I was telling you earlier that only SDN3 is classified as genetically modified organisms. Since this genetically editing mutants created to SDN1 and SDN2 techniques are free from foreign genes. I mean, no foreign gene has been included in that particular DNA. Hence, these are not classified as the genetically modified organism. So this variety that can be scheduled to release into the environment will not have, if at all, will not have any issues related to the GEAC committee under the Ministry of Environment, Forest and Climate Change. Okay. Now coming to the exact difference in all these three. See, in all the three there will be a DNA break. Okay, because only after doing the DNA break, the other technologies will follow. So, are we going to repair the template? No, in SD1. In SDN2, yes, we are going to do, but it is very short and precise. But in SDN3, it is, yes, it is a very long, includes full genes or sequences. Repair mechanism that is being followed here is NHEJ, which means non-homologous end joint. It means that no need of any inclusion of any template. Here it is HDR, here it is also HDR. What it means, the HDR? HDR is homology directed repair, where a template is inserted. And what is the edit type? Here indels. Indels in the sense insertions or deletions only. Okay. Here we are going to insert or delete from that particular thing only. But in SDN2, we are going to substitute or short sequence change we are going to make. I was telling you about the sequence change, no? Either it is from A to G or it is from C to T or the otherwise. Okay. Here in SDN3, there will be an insertion of large foreign DNA. And what is the example? See, what happens here when a gene is edited and the edit type will be SDN1. What happens here in SDN1, it is actually a process of disability or inactivating a specific gene in that particular sequence. So that is known as the indels. So what happens in this indels? It will cause a thing called frame shift mutation or a premature stop codon. That means it will prevent the gene from producing a functional protein that has been cut down. Okay, so that protein will be eliminated from that particular DNA, so that a desired result of what has been thought will be achieved. So it is known as a lockout of disease gene by a frame shift mutation or a premature stop codon, which will eventually prevent a gene from producing a functional protein that has been cut off. Here in SDN2, what happens here? We are going to correct a point mutation. For example, in sickle cell anemia, I'm just taking an example. Or in SDN2, what we will do? We will be altering the sequence. As I already told you, from A to G or C to T, we will be just altering the sequence so that the mutation doesn't happen. So the further disease or further any casualties, further disease can be prevented. In SDN3, what happens? We are going to insert a new bacterial gene into that particular crop for pest resistance. Here I am just taking the example of the BT cotton, where we are going to insert a bacterial gene into that particular, because a foreign gene has been included in that particular DNA, so that the entire structure is modified for our larger benefit. Okay. And the next point, are we going to use any foreign DNA use? No, in the case of SDN1, no, or minimal in the case of SDN2, and yes, it is typically yes in the SDN3. GMO classification, it is often non-GMO. This is also often non-GMO, but SDN3 is a typical range of a GMO. But sometimes what happens, a minimal foreign gene will be inserted in some SDN2 techniques, so that will be classified as GMO in certain countries like Germany, USA. Okay. And what are the applications? See, the main application of this SDN1 technology is to disrupt the gene. That means the turning of the gene. But here what happens in this SDN2, a precise gene correction. We are going to correct a gene so that a disease from that particular gene will not be evolved anymore. Okay, here introducing a new trait in the SDN1 and SDN2, we are not going to introduce any new trait, rather we are going to alter. But in this SDN3 we are going to introduce a new trait so that the entire DNA structure of that particular organism is altered. I hope the difference between SDN1, that is site directed nuclease 1, site directed nuclease 2 and site directed nuclease 3 are clear to you now. Now coming to the difference between the gene editing and GMO. Okay, see here, targeted modification has been done on an organism DNA, whereas an organism's DNA is altered including a foreign gene. So that is the GMO and gene editing. Here there are many techniques we are using: CRISPR, Cas9, TALENs, ZFN, and here transgenesis, CRISPR, Cas9, recombinant DNA technologies are the commonly used genetically techniques in this genetically modified organisms. DNA origin, usually there will be no foreign DNA unless with some exceptions in SDN2 type, and it often includes foreign DNA from different species. This is a very important point which you have to note it down. Precision, it is highly specific and target. This may involve random insertion or less targeted changes. And what is the purpose? It is to modify or disable a specific gene in a particular plant. Here it is to introduce a new trait. See, the main difference is that we are going to just modify or disable a specific gene that is already present in a organism. For example, we are talking about rice. Here we are going to include a new trait into that particular organism which is not at all present inherently. Okay, example, here gene edited mushrooms that don't brown. Here what they would have done, they would have just cut down the enzyme that causes browning reaction in that particular mushroom. And here we are going to insert a new trait, that is BT cotton, golden rice, everything. Okay, regulation, it is less strictly regulated in some countries. It is often subjected to strict regulations because GMO, we do not know exactly what the boon and bane of the GMOs are. And public perception, sometimes this is seen more natural, and this often face concerns or skepticism because we are not yet arrived with a concrete evidence of the boon or bane of the genetically modified organisms. I hope the difference between the gene editing and the GMO is clear to you now, and I also hope the entire lecture was clearful and useful to you. Let me sum up very quickly regarding the entire lecture which we have been going through. Till now we were talking about the newly introduced varieties of gene edited rice varieties in India, and we have seen what are the enzymes that has been targeted, and we have seen the basics of CRISPR Cas9 and CRISPR Cas12 that has been used in this particular gene editing methods. And we have come across the types of gene editing also, and we have also studied about the difference between all these gene editing methods. We have also come up across certain words like guide RNA, template, and we have also finally had a discussion on the differences between the gene editing and GMO. Now it is time for you to practice, and if you have been following the lecture till now, you will be now in a better position to answer the following practice questions. So please let me know your answers in the comment section below. Which of the following scenarios best demonstrate the use of CRISPR-Cas9 improving drought resistance in crop species? Option A, inserting bacterial gene into maize to improve water use efficiency. Option B, silencing a native gene in rice that negatively regulates drought tolerance using CRISPR-Cas9. Option C, crossbreeding two rice varieties to introduce a drought resistant traits. Option D, applying chemical treatments to plant during water stress conditions. So let me know your answer in the comment section below. Moving to question number 2, what is the major advantage of using SDN, site directed nuclease 1 genome editing in crop plants? Option A, it introduced foreign DNA to improve plant immunity. Option B, it allows precise point mutation without introducing foreign DNA, simplifying regulatory approval. Option C, it is a slower process compared to the traditional mutagenesis. And option D, it always requires a selectable marker gene for plant regeneration. So please let me know your answers in the comment section below. And the last question, India recently approved genome edited rice varieties like DRR, Dhan 100 and Pusa DST rice 1. Which of the following best describes the distinguishing features of these varieties and their regulatory classification under the India Biosafety Guidelines? They are classified as GMOs under the India's Environmental Protection Acts and require full biosafety clearance before release. They were developed using CRISPR-Cas9 and SDN2 editing to insert foreign DNA for enhanced drought resistance and yield. They are SD1 gene edited crops with no foreign DNA and are exempted from GMO regulation under India's 2022 notification by DBT. They rely on RNA interference to silence stress response genes and are subjected to same regulatory framework as BT crops. Kindly go through the question again and Aram say let me know your answers in the comment section. Okay. So I hope this session was useful to you and this topic becomes very important from your both NABARD grade A and IBBS AFO 2025 examination. So if you are looking for a good test series for your upcoming IBBS AFO 24/7 IBBS AFO examination, do consider our test series, that is the IBBS AFO 24/7 test series by Clarity, where we are providing tests based on block wise. Along with the test, we are also providing notes for quick revision. So if you are looking for a good and affordable test series, do check the link in the description below. And if you're looking for a good mentorship program for NABARD, Clarity for sure uh, is giving you a NABARD mentorship and test series program for 2025. We have given a tremendous result for the past 3 years, and we will be for sure replicating that in the 2025 cycle also. So if you're looking for a good test series and an affordable test series for NABARD mentorship also, do consider enrolling our test series, for which the link is given in the description box. So finally, I hope the session was useful to you and the session was informative. If you have any feedback related to the session that has been held, please let me know your comments in the thoughts in the comment section below. So thank you so much for watching this video, and I will meet you all with yet another session. Till then, happy learning. Have a good day.